she too, after training at the NCI during her fellowship and focusing on GI and GU cancers, she's opted to focus on drug development within the FDA. And it's always enlightening to hear how our government leaders are having an impact on moving the bar forward. And there's I think, often a misconception that the FDA is a sort of regulatory, it is, but what they're really trying to do is break through, is to try and change the standard of care. But they need to do so responsibly and appropriately to make sure that on the backs of everybody, we move it forward better. So with that, I look forward to her comments on how she's going to move things forward. Dr. Gandhi.
Okay. There you go. Thank you, everyone. Can you hear me? Okay. Thank you, Aki and team, for having me here. I am glad to talk to you all about the FDA endpoints and the clinical trial design and all the things we consider when approving a drug. My name is Shruti Gandhi. I'm a medical oncologist, a GI medical oncologist. I work at the FDA in the GI oncology division. And I'm going to talk to you a little bit about what we do at the FDA and sort of how we overall regulate drug approvals, and give you some specific examples for GI cancers and gastric cancers. I have no disclosures. When I was a fellow, and I was a fellow at NCI, I was quite surprised to know that I actually didn't know a lot about what the FDA did when it came to drug regulations and how some of the drugs that I was actually using in clinic were approved. And so I feel like it's always been good whenever I talk to patients to sort of give them a brief overview of the FDA and the Oncology Center of Excellence, which is what I work under, and how we go about approving drugs and what our general mission is. So this is my brief outline. Then I'll talk a little bit about drug development, cancer drug development, and the clinical trial endpoints we use. And then lastly, I'll end with the approval pathways. And I think there's been a lot of talk in the media about a lot of the drugs that we've approved recently. I do want to point out that I am an oncologist, and while the FDA and other divisions do work within the same sort of regulatory framework, my focus has been on oncology, and I'm actually not privy to the discussions that happen in the infectious disease group. And so it's a very, very engaging environment. But I'm not always included in all the discussions for other divisions. So at the mission of the FDA, what we try to do is we assure the safety, efficacy, and security of drug biological products, also medical devices, food supply, radiation products. As Dr. Marshall mentioned earlier in this talk, we cannot take into account cost or payment issues, and we do not regulate the practice of medicine. So what that means is, while we do understand cost is important, it's actually not something within our regulatory legislative purview. We do not know the cost, the future cost of the drug when we're undergoing the review and approving it. So that's something that's completely sort of hidden from us. We're just not, that's not something we take into consideration at all. So the safety and efficacy requirements, the laws which we work under, the Food and Drug Cosmetics Act was first actually enacted in 1930s and then amended in 1960s. And it basically says that the drugs that are approved for marketing must be found safe and effective. And it was amended to also say that, generally we want to have well-controlled clinical trials to show that these drugs are safe and effective for patients. For biologics, there was a similar act called the Public Health Services Act, and both of them sort of work under the same evidentiary framework, these are the legal sort of guidelines which we have to work under, and this is what we use as our basis for drug approvals. So I work under the umbrella of the Oncology Center of Excellence, and a lot of people don't know this, but it's an intercenter institute. We actually interact with other centers. So I work under the Center for Drug Evaluation and Research, which is CDER, but we also work with Center for Biologics Evaluation and Research and Center for Devices and Radiological Health. So we sort of work very closely. We leverage the expertise of physicians, scientists, pharmacologists, toxicologists, chemists, and who have expertise in different areas. So it's a great, and statisticians, of course, and it's a great, very interactive environment. Within the oncologic group, we have three divisions that are focused on solid tumors and two on heme malignancies. And so I'll talk a little bit about the cancer drug development. Some of the challenges that we see as oncology drug reviewers really are unique to oncology drugs because they treat life threatening diseases. We do have to take into consideration and provide a balance between patient access and then also making sure that the drugs are adequately investigated, the drugs or biologic agents. Um, Again, as Dr. Marshall mentioned earlier, we are having smaller and smaller trials, mostly because a lot of them are biomarker driven, or they're in rare diseases, which we really encourage. But these trials also involve smaller population of patients, a shorter exposure time. So we have to take that into consideration. They might be single arm trials. And we take all of these things into consideration when we're saying, are these drugs safe and effective? Um, in the past, we've also actually accepted historically severe toxicities because if a drug has been proven to be efficacious. And I think our thinking a lot is changing on that, especially because we hear back a lot from patients saying, no, a lot of drug toxicities are not acceptable. Things that we used to we used to say are manageable or controllable with medications, for example, diarrhea, nausea, vomiting. We sort of want to make sure that the drug is now optimized, the drug dosage is optimized so that we can minimize those toxicities while providing the same level of efficacy. And also, of course, risks and benefits change with different types of cancer. Within a certain type of cancer, they can change based on the stage of cancer. So that's also something we have to take into consideration when looking at the review process. And lastly, there are ethical concerns that we grapple with when it comes to trial design. Like I said, we were historically looking at well-controlled, randomized trials to look at drugs and make sure that they were safe and efficacious. However, we have in the past few years, especially, approved a lot of drugs based on single-arm trials. And especially if these trials show that the drug is efficacious, if it has tolerable toxicity, if it doesn't show any difference within relevant subgroups, it would actually be unethical for us to ask the sponsor, the drug company, to run a randomized trial because we know this drug works, we know it's helping patients. So that's something that we actually really strongly take into consideration. Another thing that we take into consideration is if a drug has very clear, very early benefit, for example, MSI high cancers and treatment with with immunotherapy has very quick, very early, and very robust, especially MSI high gastric cancer. We cannot. We would not want to require a randomized clinical trial in some cases when we know that there is very clear benefit. Also, another thing that we grapple with is that observations in clinical trials don't really predict real world experience. It is in clinical trials difficult, but also in real world to see whether or not the adverse events that we see are due to the drug, due to the disease, or both. In clinical trials, we have seen, again, historically, they're excluding patients who would normally get this drug based on the clinician's judgment in the real world. So, for example, clinical trials usually only enroll patients who have really good performance status. They exclude patients who have brain metastases, leptomeningeal disease. They also exclude patients who have poor kidney function, poor liver function, and we're seeing that in the real world, the applicability of these parameters is not consistent. So a lot of clinicians will give these drugs to patients if, in their judgment, they think patients can tolerate. So that's another challenge, and we are actually actively working to expand eligibility criteria so that more patients, for example, if they are fit, but if they have brain metastases, or if they're fit but they have peritoneal disease, they they should be enrolled in trial if it's safe for them to be enrolled. So we're trying to work. We have guidances out, and we're actively trying to push to make sure that patients are being enrolled. Um, Lastly, this is another thing that we work with. So, inadequate demographic representation and healthcare quality are both very different. With regard to demographic representation, we often see that sometimes older patients are excluded from trials. And more often than not, and we're trying to again correct this through one of our projects, the patient population in the trial doesn't reflect the US patient population that's going to get the drug. So we're really pushing to get more racial and ethnic minorities enrolled in these clinical trials, especially because first of all, they respond very differently to drugs. They they can have different side effects. They can have different efficacy, and we want to make sure that the patients in the U.S. will be getting it. These drugs are adequately represented in clinical trials. So, and lastly, of course, frequent off-label use. Again, we do not regulate the practice of medicine, so there is frequent off-label use for a lot of drugs, and that is something that's different between clinical trials and the real world. So, all in all, we do have to strike a balance. We want to make sure that we require adequate amount of certainty and data, but we don't put too much regulatory burden because. When we do want if we require a lot of certainty, we require a lot of data, a lot of times that can be stifling innovation. It's actually going to make provide more regulatory burden to the sponsors, and it's not going to have the drug available to the patients who need it as quickly as possible. However, we also want to make sure that we are paying attention to the safety findings. We're making sure that the toxicity of the drug is well reported across the trial, and that's something that we really continue to monitor even after the drug is approved. So now I'll talk a little bit about clinical trial endpoints. Really, again, this differs from cancer to cancer, and even within the cancer it's quite different, but generally we look at safety and efficacy to determine if the risk benefit that a drug treatment provides to patients is acceptable. So with safety, we usually measure it with adverse events that we see in clinical trials as reported by investigators. And again, here we have some issues with single arm data where we see patients who are in single arm trials, but we don't know, because there is no control arm, we don't know if the adverse events they're experiencing are due to the drug, due to the disease, or both. But it's very sort of hard to tease it out. And again, toxicity, we've generally accepted higher drug toxicity in oncology due to higher mortality rates. But we have another ongoing project called Project Optimus that really is working with the drug companies even before they start the registrational trials, before they start these trials, enrolling patients to make sure that the drug dose that they're giving has been optimized to minimize toxicity and optimize the efficacy. So that's something we really want to do. We also measure efficacy using clinical trial endpoints, which I'll talk about in a little bit. But in general, we want to answer the question, does the product provide meaningful clinical benefit to patients? And that's really something we look at in our review. So the kind of questions we ask during clinical trial efficacy evaluation are very different during the early phase and late phase. So, for investigators, such as Dr. Taylor, we see a lot of investigational new drug applications looking at different therapies. And I actually personally, having trained at the NCI, I think this is really exciting. This is where a lot of the innovation happens. And we really try to work with these investigators. These are usually smaller trials looking at new therapy either alone or in combination with standard of care. But we want to know if the drug is actually active in that case, and that's very early. And our goal when we review these applications is to make sure patient safety is accounted for, because we want to make sure that patients are aware of the toxicities of the drug as far as they're known. And we want to make sure that patients know that if they're going to be getting a drug or a drug combination, they might be foregoing standard of care, for example, if they want to enroll in a clinical trial. So those are some of the things we always want to ensure that patients are informed of in the informed consent form, and that's something we look at very thoroughly in the early phase of development. Um, the late phase of development, we usually want to look and see if the drug provides meaningful clinical benefit. And that's something that's, we talk to sponsors and pharmaceutical companies before they start the trial to make sure an adequate control arm is used, where they're not using something that's antiquated, they're not using a arm that's old, that patients are not getting anymore. And then during the approval process and post marketing, which is the new drug application and the biologics application, we look to make sure that safety is being continually monitored, especially for things like immune checkpoint inhibitors that can have long term safety side effects, not just 30 days after the patient has stopped the drug, but those side effects can show up a lot later, 90 days after. So we want to make sure that those drugs are and those safety events are being recorded. So we're actually working continually and evaluating the data we're getting continually from these drug companies and sponsors to make sure that the drug is actually safe. And let's see. All right. So some of the things we measure and look at when we're approving a drug, we look at direct measures. So of clinical benefit, this is really looking at how patients feel, function, and survive. And historically, we've looked at overall survival, which is really how long does the patient live when they're given the drug. These days, we're also putting a lot of attention on clinical outcome assessments, and I'll talk a little bit more about that in a bit. Some of the indirect measures we look at are measures that kind of predict clinical benefit. And these don't actually are not in and of themselves something that you would use to approve a drug, but things like disease-free survival, in some cases response rate, overall response rate, or complete response. Those are things we would use to predict, hey, is the patient going to live longer? Are they going to do better when they get this drug? And a lot of them in a lot of different disease groups are quite established. And we're also sort of working to see what other intermediate endpoints we can use to get the drug, to get it approved faster, to get it out to the patient if it works. And I'll talk a little bit more about that in a bit too. Oops. Yeah. All right. So clinical outcomes assessments, that's something that, especially patient reported outcomes, is something we're taking into consideration quite a bit. This has its pros and cons because, first of all, it really tells us how the patient is feeling. Things like diarrhea, vomiting, it's something that the patient can report and say, we're getting X number of episodes of diarrhea. The vomiting is, we're vomiting a lot all day, and it's something that's measurable, and we can actually do something about it. There are some other non-observable concepts such as pain and itching that only the patient can tell us. So one of the downsides with this is, one of the limitations, I should say, is that it's quite hard to find consistent assessment and metrics. There are quality of life measurements available. It's difficult to do. A patient who's out there getting chemotherapy treatment, any kind of treatment, it's hard to go out and fill a form while you're waiting. There's also recall bias, and a lot of times patients cannot remember how they felt 2 days ago, how they felt 5 days ago. It's how many episodes of diarrhea, how many episodes of vomiting they had. So there are certain limitations, but we are taking into consideration patient-related outcomes, especially when they contribute overall to the quality of life of the patient. Um, and some of the indirect measures, and I won't spend too much time on this, but response rate and progression free survival are two things that we look at as sort of intermediate endpoints. And for example, in gastric cancer, we have approved Keynote 811, we approved Pembro, trastuzumab, and chemotherapy based on a response rate alone, and because that showed us that this was probably indicative of a survival benefit. And as I'll talk about later, it is incumbent on the sponsors, it's incumbent upon the drug company to provide us ongoing evidence, and confirm that the clinical benefit is there if we use an indirect measure of efficacy. So to summarize, we use direct measures, established indirect measures, as well as some unestablished indirect measures. And then I'll talk about approval pathways, which I think is something that a lot of people are interested in, especially a lot of oncologists, because we do tend to approve a lot of things these days with accelerated approval. So we have two approval pathways: regular approval and an accelerated approval pathways. And it's interesting because the regular approval is what had been historically used to show that the drug had substantial evidence of safety and efficacy. Again, we wanted to have 2 or more well-controlled clinical trials, and it's really based on a direct measure of clinical benefit. Importantly, when a drug or when a drug company applies for a regular approval, they just have to show that the drug is as safe and as effective as existing therapy. So a good example of that would be in HER2 or famtrastuzumab deruxtecan. It was approved in a later line setting for patients with gastric cancer, and it was given a regular approval based on comparison with chemotherapy. So it allows for this kind of an approval, allows for non-inferiority design, and it's really one of the standard mechanisms. Sorry about that font, but accelerated approval is something that we've used more and more recently. A little bit of a history. It was actually sort of implemented back in the 80s to approve and provide drugs to patients during the HIV crisis, to actually expedite the review of drugs and approval of drugs during the HIV crisis. Now we use it frequently in oncology because it also requires substantial evidence of safety and efficacy, but we can use it we can approve a drug with accelerated approval based on an indirect measure such as response rate or progression-free survival. So any endpoint that is reasonably likely to predict clinical benefit. One of the pros and one of the reasons we really like this is because it provides for earlier events, smaller, quicker trials. But as I said, it is a requirement and a lot of times sponsors, pharmaceutical companies have a post-marketing requirement to show that this actually has clinical benefit, to confirm that. So they have to continue providing us data. And again, for the Keynote-811 trial, which approved pembro, trastuzumab, and chemotherapy, we are actually requiring the sponsor to provide us with ongoing data to make sure that patients are having a survival benefit with this treatment. So that's something we look at. And another thing is that it must demonstrate benefit over a currently available therapy. So any drug that applies for any sponsor that applies for an accelerated approval for a drug must show that the drug is going to be beneficial than whatever is available. And again, this was in place mostly to expedite the the availability of these drugs to patients who need it most, especially if we think that this is going to help patients. All right, so this is actually just my conclusion. I have a couple of conclusion slides, but I just really went over very quickly about the review of cancer therapeutics and how it involves serious life-threatening diseases. And it is a really active area. The kind of applications we get, it's really the cadence is determined on what's being done, how active the pharmaceutical companies are within a certain disease group. We obviously try to get as many drug companies to, we work with them to get as many trials as we can. But again, this depends on the scientific the landscape as well. We look at the safety and efficacy data to look at and examine and assess for meaningful clinical benefit. We look at direct measures, and especially we're paying more attention to patient-reported outcomes now. How do patients feel, function, and survive? And then we also look at indirect measures to predict clinical benefit. Regular approval, as I talked about, is normally based on measures of clinical benefit itself. And then accelerated approvals are based on intermediate endpoints and often require a confirmatory trial. So ultimately, the appropriateness of a particular trial and endpoints really depends on the disease, the stage, the availability, and the scientific landscape of that disease. So we really work very closely with sponsors as they submit us these clinical trial designs to make sure that it's applicable to the US patient population. And then we also have resources, and we engage quite a bit with patients at the FDA. We have patient listening sessions, and I will end my talk there. Thank you for, yeah.
Thank you.